So, good afternoon everyone and welcome to the Ph.D. defense of Chandni Shah. We have two eminent teachers and scientists with us today. First of all, I welcome Professor Pradeep Verma, who is a professor in the Department of Microbiology at Central University of Rajasthan, Ajmer. He did his PhD with Professor Datta Madambar from our department, and now he is a very senior faculty in this department. He is also dean of the faculty there. We are very happy that he has spared his valuable time to attend this course. Thank you, sir. We also have an equally eminent and well-known microbiologist with us, Dr. Chandni Shah. She is professor and head department of microbiology at Patel University, Ahmedabad. Madam is also working on a similar area. So she and Dr. Pradeep, these two were actually the right examiners for evaluating the thesis of Chandni. I am happy that we have got the right experts. I welcome you also, madam, for the vivas. Ma'am is attending this viva as an observer. Both were examiners, and he is selected as an external and ma'am is an observer. So all three examiners are there. That's a good thing. I welcome all the other colleagues and students and participants. And with this brief introduction, shall we start this, please?
Good morning to one and all present here, and thank you very much for coming to my PhD defense. Today, I will speak about my PhD work entitled as Biochemical and Molecular Studies on the Role of Microflora on Phosphate Uptake in Banana. The entire work was conducted under the supervision of Professor R.B. Subramaniam at PG Department of Biosciences, Patel University. So I will start with a brief introduction. Bananas. Bananas are a nutrient-rich, long cylindrical berry fruit. They are free from cholesterol, sodium and fat. Apart from their nutrient values, they are cultivated in more than 120 countries all around the world. From all, India is the leading producer and exporter of the bananas worldwide. And state-wise, Gujarat stands second in the production after Tamil Nadu. As we all know, after the Green Revolution, most plants' nutrient demands were mainly fulfilled by applying chemical fertilizers. And banana cultivation demands large amount of fertilizers, specifically nitrogen, phosphorus and potassium-based fertilizers as well as pesticides. From all, phosphorus is an essential macronutrient for the growth and development of all the living organisms, and they play significant role in the plant growth. According to the Food and Agriculture Organization, approximately 30 to 50 kg per hectare of phosphatic fertilizers are applied annually in India only. So surprisingly, plants take up only a tiny fraction of 0.2%, and the rest are added into the soil as an excess phosphate. So based on the literature study, approximately 5 million tons of phosphatic fertilizers is being excess in the year of 2019 to 21. This was quantified by observing the difference between the input of fertilizers and the output of amount of crop harvested. This rest of the phosphate is lost into the soil and creates a pollution by damaging the soil microbial biodiversity, leaching out into the water bodies and creating a situation of eutrophication and also converted as an insoluble phosphate. Here is the schematic in and outflow of phosphorus cycling. Two types of phosphatic fertilizers, organic and inorganic, are applied and added into the soil. Plant absorbs a small portion and rest is converted into insoluble phosphate through immobilization, absorption and precipitation. The present existed rhizospheric microorganisms have ability to convert this insoluble phosphate into soluble phosphate through mineralization, desorption and solubilization. But due to over exploitation of agrochemicals, rhizospheric microorganisms also hamper and lost their presence. Here is the general mechanism how this synergistic interaction between plant were promoting rhizobacteria like mycorrhizal fungi and phosphate solubilizing bacteria help in phosphate uptake. So plant root release root exudates which contains amino acids on sugars for the growth at the zone of rhizosphere. And this PGPR organism take up these root exudates from the growth for their growth and in return provide mineral nutrients like phosphorus. In that, phosphate solubilizing microorganism solubilize the inorganic and organic insoluble phosphate into soluble plant uptakeable phosphate by releasing organic acids, enzymes, hydroxyl or carboxyl ions. This soluble solubilized phosphates are taken up through the mycorrhizal fungi via Ph.D. 1 and 2 transporters in the form of orthophosphates and polyphosphates and ultimately provided to the plant. But due to under certain circumstances like nutrient deprived conditions and microorganism resistant conditions, plant under the stress condition, plant release several PR related proteins and signaling molecules such as NSL, homocysteine, lactate, mimicates and stereogalactins, which were recognized through a group of bacterium present in the soil. This mimicates triggers the proliferation and expansion of other bacteria known as mycorrhizal helper bacteria. Thus, this proliferation of mycorrhizal helper bacteria releases the enzymes and interaction molecule which express several genes to trigger the sporulation of mycorrhizal fungi, release of exopolysaccharides, enzymes, and increasing an interaction and proliferation of other vegetable organisms. Hence, already available mycorrhizal fungus spores also proliferate and facilitate the inorganic soluble phosphate into the plant. In return, plant provide more amount of carbon-rich nutrients to the organism. So, based on the literature study, the objective of the present study is to identify the potential phosphate solubilizing bacteria, mycorrhizal helper bacteria, and their interaction with arbuscular mycorrhizal fungi to alleviate the phosphate deficiency in banana. So the first objective of the proposed study is the isolation and identification of phosphate solubilizing bacteria, their plant growth promoting properties on banana. Here are some of the sub-objectives. The graphical representation shows the isolation and screening of potential phosphate solubilizing bacteria from the banana rhizosphere, identifying characterized for their PGPR properties, determine their plant growth promoting properties on banana field, and lastly, best of the PSB's genomic characteristics were identified by performing whole genome sequencing. So site we are selected from Anand, Gujarat, and rhizospheric soil was collected from the banana rhizosphere. Their physicochemical properties of the soil were analyzed. Based on the soil analysis, pH is alkaline in nature, total organic carbon and nitrogen is very low, where macro and micronutrient content is moderately low. From the soil, total 7 phosphate solubilizing bacteria were identified and evaluated for the inorganic and organic insoluble phosphate solubilization. Here, organic as tricalcium phosphate and inorganic as sodium phytate was selected as a phosphate source because after the continuous application of fertilizers and pesticides, the other agrochemical majority of the global soil is converted into into a calcareous or alkaline in nature. And reports represented that under the alkaline condition, applied phosphate has a significant tendency to bind with calcium ions than the iron and aluminium. Thus, here we selected tricalcium phosphate. Meanwhile, sodium phytate is one of the readily convertible form of applied phosphate, and several living organisms also react with available phosphate to convert them as a phytate. So, from the 7, 3 are the most phosphate solubilizers were selected, I3, 4 and 7, and renamed as PSD 01, 2 and 3 respectively. Then 3 of the isolate soluble phosphate content and pH of the medium was recorded. Their correlation analysis was performed. According to the literature survey, increasing in the soluble phosphate content is directly negatively correlated with the decrease in the pH of the medium. And our study also shows the same and observed significant negative correlation between them. Then the mechanism behind phosphate solubilization was also determined by identifying the presence of organic acids and enzymes. Results shows that 3 of the isolate PSB 1, 2 and 3 release the gluconic, citric, lactic and salicylic acid, and their presence was confirmed by performing HPLC. From all, PSB03 released highest acid phosphatase and PSB03 is the lowest. The results are vice versa for the alkaline phosphatase. Beside organic acid production, phosphate solubilization is also carried out through carbon metabolism, enzymes, siderophore, hydroxyl or carboxyl ion release, through protonation, respiration and extrusion. Further, 3 of the identified isolates molecular identification was carried out by performing 16S rRNA gene sequencing, and based on NCBI gene bank database, PSB03 1 and 2 belongs to Bacillus species, where PSB03 belongs to Staphylococcus species. 3 of the isolate sequence were submitted to NCBI to obtain their accession numbers, and their phylogenetic tree was constructed by using neighborhood joining method in MEGA10. Then their various biochemical and PGPR characteristics were identified, and result shows that three of the isolate produce lipase and catalase enzymes and can be considered as PGPR, as they show the production of IAA, ammonia, siderophore, HCN, H2S and phosphate. According to the previous study, phosphate solubilization also governs through the release of acids and enzyme production, and their survival in the field also depend upon the carbohydrate utilization. In the present study, 35 different carbohydrates were considered for their utilization assay. Based on the result, PhD03 utilized 15 different carbohydrates followed by PhD1 and 2. So, based on the previous studies, biochemical and PGPR characteristics, PhD1 and 3 are the most proficient, and we have selected further to determine their plant growth promoting activity on banana at the field level. Here is the general methodology for inoculum preparation and inoculation into banana plant as a different treatments to study their morphological and biochemical characteristic till 90 days. After 90 days of incubation, plant morphological and biochemical parameters results shows that T3, which contains PhD03, shows the significant improvement in plant height, number of leaves, as well as biochemical parameters. So, as per the results of field study, PhD03 finds the most effective. Thus, PhD03 is further selected for its in-depth genomic characteristics by performing a whole genome sequencing. Here is the general workflow of next generation sequencing from DNA extraction, library preparation, sequencing, and analysis. PhD03's genomic characteristics were identified by performing whole genome sequencing using Illumina MiSeq platform. Obtained genes were annotated using de novo assembly, and genomes were annotated using NCBI PK annotation. Project were submitted to NCBI to obtain the accession number. The RAS subsystem coverage were also visualized, and genes and their metabolic pathways were confirmed through cake pathway. Based on the NGS data analysis and literature survey, we also identified that following isolate did not have any active virulence factor directly involved in pathogenesis as OSS. These are the list of genes involved in phosphate solubilization, IA and plant hormone production, ion sequestration, and nitrogen metabolism. And these are the pathways of nitrogen metabolism and inositol phosphate metabolism, as well as fructose and mannose metabolism. Highlighted green color shows the genes present in PhD03 isolate. The conclusion of chapter 2 is 3 out of 7 PhDs were selected for further biochemical and PGPR characterization and identified as Bacillus species and Staphylococcus species. The isolate Staphylococcus species PhD03 is the first time reported as a PGPR, which showed the potential to utilize 15 different carbon sources. Plant growth promotion study also shows that PhD03 is the most effective by improving the morphological and biochemical parameters of the banana plant. Whole genome sequencing and genome annotation of PhD03 also shows the presence of various PGPR related genes such as inorganic and organic phosphate solubilization, ion sequestration, IAA production, and nitrogen metabolism. The tentative pathways were also identified by cake analysis. So, in chapter 3 is combined inoculation of previously identified PhD03 with arbuscular mycorrhizal fungi to alleviate the phosphate deficiency in banana plant. In this schematic, PhD03 were collected and studied their effect on AMF spore germination under in vitro monoxenic culture method. Followed by their study their impact on banana plant and their correlation analysis between plant growth promoting parameters as well as soil parameters. Here is the general life cycle of AMF under 40x, observed at 40x. And by performing monoxenic overlaid agar method, AMF spores were germinated in the presence and absence of PhD03. Results showed that in the presence of PhD03, 85% colonization was observed compared to the control. Then the molecular identification of mycorrhizal fungi was initially carried out by performing 18S ITS sequence amplification, followed by glomus sequence amplification using glomus gene specific primers, and last results showed 100% identity with glomus proliferum. Their sequence were submitted to NCBI to obtain the accession numbers. Then both PhD03, AMF, and their combined inoculum were prepared and added into the plant to study their growth enhancement effect on banana at a of incubation, treated plants show drastic improvement compared with the control. Moreover, treated plant also shows increased mycorrhizal colonization, plant height compared to the control, and root protein content of this also improved. So, we also analyzed the physicochemical analysis of treated rhizospheric soil. And the rhizospheric soil analysis also suggested the prepared combination of Ph.D. and AMF positively improve the total organic carbon, total nitrogen and phosphate content. Moreover, mycorrhizal spore numbers are also improved fourfold improvement compared to the control. Lastly, correlation analysis was performed by using all the plant and soil parameters, and results showed negative correlation with only soil pH, while all the other parameters are positively correlated with each other due to production of organic acid and active stage of bacteria. The conclusion of chapter 3 is AMF identified as global species. The sequence we have submitted and obtained accession number. In vitro germination study shows that combined inoculations show 85% higher colonization. Based on plant study, combined inoculations shows the most significant effect and enhancing the morphological and biochemical parameters of the banana plant. Treated rhizospheric soil also show improved content of total organic carbon, total nitrogen and phosphorus. So the chapter 4 is isolation and identification of mycorrhizal helper bacteria and their effect on their effect on growth of banana plant with AMF and Ph.D. In the fourth chapter, mycorrhizal helper bacteria, we are isolated from the mycorrhizal inoculated plant of previous chapter T3 and T4 by collecting the plant roots and rhizospheric soil, determine their PGPR active properties, and lastly prepared a bioformulation using Ph.D. and AMF and studied their growth promoting effect at a field level. Here is the graphical representation of this same. For the isolation of mycorrhizal helper bacteria, initially mycorrhizal spores were collected by performing wet sieving and decanting methodology. With the help of stereo microscope, 14 different globulous to subglobulous shaped yellow to brown color spores were collected and inoculated into a bacterial growth medium. Moreover, mycorrhizospheric soil suspension is also prepared and inoculated into the growth medium. A total of 65 morphologically distinct MHB isolates was collected from this. From this, 1 to 21 are from the spores of the fungi and the rest are from the soil. All the isolates preliminary screening was performed through their biofilm formation, swivel motility, production of protease enzyme and release of MHL type sensing molecules. Of all, 12 isolates shows all the properties. We are selected for further morphological molecular identification and biochemical and PGPR characterization. So from all 12 MHB isolates, MHB4CP is the most potential, identified as a gram-negative rod-shaped, have solubilized phosphate and release lytic enzymes such as amylase, lipase, catalase and gelatinase are selected. All the isolates we are identified through 16S RNA gene sequence. Their sequence we are also submitted, and MHB4CP belongs to Enterobacter propane family. Then this MHB4CP isolates physical association with mycorrhizal fungi was confirmed by performing FDG-SEM analysis on banana leaf after 60 days of incubation under sterile condition. From the micrograph, the red color arrow shows the presence of arbusculin spores of the fungi in T2 where the only AMF was added, and yellow color shows the colonization of bacterium in T3 alone, and combined inoculation of T4 where mycorrhizal fungus and MHB4 added was observed the physical association of both bacteria and fungi. After the identification study, both the identified isolate Ph.D. from the chapter 2 and MHB4CP cities exopolysaccharide production was performed. EPS production plays a significant role in the biofertilizer preparation, and these are the major characteristics of EPS production. Both the bacterial isolates exopolysaccharide production property was evaluated using 6 different growth medium for 7 and 14 days. Results represented that PSB shows highest EPS production in basal minimum medium, while MHB shows highest in Duria verdonis blonde. Later on media we are also optimized by following one factor at a time condition and for both the isolates here. Total 6 different carbon and nitrogen sources we are considering. From all, both the isolates show significantly enhanced production in the presence of glucose and urea. Meanwhile, physicochemical characteristics were also analyzed, and result shows that at pH 7, temperature 30 degree, aeration at 100 rpm, and increased inoculum percent of 3% is the most effective for both the isolates to increase the EPS production by twofold. Then beside EPS production, isolates and their combination of PSB, MHB and PSB plus MHB were considered to determine their capability for bioremediation of pesticides. Here 8 different well-known pesticides were considered, and degradation study showed that combination of PSB and MHB is the most effective and utilize and bioremediate several pesticides. From all the 8 pesticides, chloropyrifos, combination of both shows the highest degradation of chloropyrifos. So we further proceed for the higher concentration till 100 mg per liter. And surprisingly, combination of both the bacterial isolate PSB and MHB shows complete biodegradation of chloropyrifos within 15 days. So after confirmation, we try to prepare a bioformulation using 3 of the identified isolates of the present study, PSB03, MHB4CP and mycorrhizal fungus. These are the primary inclusion criteria behind the selection of carrier material. Here we selected wheat straw and soil as a bacterial carrier material. These are the physicochemical properties of the bioformulation before and after bacterial inoculation. After bacterial inoculation, water holding capacity, moisture content and total phosphate content is increased. Apart from this, second most important parameter is the shelf life of the living organism in the carrier material. The shelf life of 3 of the organisms is also determined at room temperature, and results showed that both the bacterium isolates retain their shelf life up to 200 CFU per gram, while fungus maintain their life till 5000 IU per gram till 80 days. Then 8 different bioformulation combinations we are prepared to study the effect at seed germination on maize and pot and field experiment on banana plant. The result shows that T7 treated seeds show significant improvement compared to all the other treatments as well as control. The treated seed and seedling show increased fresh and dry weight and root and shoot length, and increasing the total phosphate content of the root compared to the similar type of results we are observed for morphological characteristics of the banana plant at both pot and field study, and recorded 6-fold enhancement compared to the control. Moreover, T8, T6, T4 also show significant improvement. Results are same with the biochemical characteristics of T7, which include MHB and AMF, and improve the carbohydrate, protein, total chlorophyll, proline, ascorbic acid content by 8-fold compared to the control. And T8, T6 and T4 also show improvement, but T5 shows the lowest improvement or no improvement compared to the control, which include only AMF. Then the plant-microbe parameter results were also hand-to-hand with the results of other biochemical pot and field experiments, and show increasement in T7 and show also improved root protein content of the T7 treated plants. Further bioformulation added soil analysis was carried out and showed that pH and EC content is decreased. At the same time, total organic carbon, total nitrogen, phosphorus, potassium and all the considered micronutrient content is increased except the iron, which is maybe due to release of pseudophore by the bacteria. Then all the pot and field study considered parameters, correlation analysis was carried out, and result shows that all the parameters were significantly positively correlated with each other except the leaf area of the pot. This is due to the limitation of the growth area, where all the field study parameters were positively correlated with each other. So the conclusion of chapter 4 is initially more than 65 MHB isolates were screened. 12 best isolates were selected for further study. Selected isolates were biochemically, morphologically and molecularly characterized and identified. Among all, MHB4CP show various PGPR characteristics. Therefore, it was selected for AMF spore germination assay. Physical association between MHB4CP and AMF was confirmed by FAG SEM. The isolate Ph.D and MHB produced exopolysaccharide and efficiently degrade chloropyrifos within 15 days. So bioformulation was prepared using isolates of PHB03, MHB4CP and AMF with wheat straw and soil. The shelf life of the bioformulation was 80 days at room temperature. Result shows that the bioformulation improved the morphological and biochemical parameters of the maize and banana plant. Among all the treatments, P7 of MHB and EMF based bioformulation significantly increased all the growth parameters of both plants compared to the control. So, chapter 5 of last objective is detection and identification of signaling molecule from MHB Enterobacter species and its effect on banana plant. Here, as per the results of the previous chapter 4, MHB and AMF inoculated plants show highest improvement. Thus, in the last chapter 5, we detected and identified the interaction molecule involved in the communication between plant-microbe interaction through various spectroscopic methods and study their effect with bacterium and fungi. Here is the graphical representation of the chapter 5. With the help of bioreporter strain CV026, all the 65 MHB isolates screened through parallel stripping method and confirmed using positive control as CV from MTCC. One of the isolates showed positive activity, were selected and their identification was carried out through polyphasic approach. Based on their detailed biochemical and PGPR characteristic and carbon utilization assay, selected isolate is also considered as phosphate solubilizer, IAA, ammonia, HCl producer and utilized 23 different carbons from total 33. The selected isolates also show fluorescence and their morphological and molecular identification was performed. Further, phylogenetic tree was constructed using MEDCA 10. The isolate belonged to Enterobacter fungi. Then, authentication of bacterial released NaHL was confirmed by using 6 different carbon chain length standard NaHLs. Here is the chemical structure of all the selected NaHLs. Again, the extraction of bacterium released NaHL was carried out by performing liquid-liquid extraction methodology and concentrated using rotaiopeter. Then, extracted NaHL separation was performed by TLC and bacterial NaHL showed 2 spots while HPLC shows the presence of C6 type NaHL molecules. So the samples detection and structural elucidation of bacterial NaHL was carried out by performing FTIR analysis with standard C6 type NaHL molecules. An obtained sample NaHL shows similar pattern with one extra CX stretching, CC stretch and amide stretch. While with the help of NMR spectroscopy, we predicted the structure and tentative structure belong to N3-oxo C6 type NaHL molecule. Further, the confirmation structure further confirmed through GCMS spectroscopy with standard and sample NaHL. And based on the NIST library, this compound belongs to N3-oxohexanoid L-homoserine lactate molecule. Then after the released NaHL, MHB bacterium, AMF and their 8 different combinations we are prepared and inoculated into the soil to identify their effect on soil microbial biodiversity. So total 8 different growth medium we are used to identify the presence of different microorganisms. And after 30 days of incubation, soil samples from all the treatment were spread on all the 8 growth medium, and result shows the highest biodiversity is found in T7, T8, while lowest in T1 as a control. Thus, the PLFA analysis was carried out. The results of PLFA analysis suggested that, in compared to control, T7 shows the increased number of microorganisms. In that, T1 shows 126.63 nanomole per gram of biomass, while T7 shows 735.78 nanomole per gram of biomass, with 6-fold improvement in the soil microbial biodiversity. Then all the prepared combination we are also used for seed germination and field application on maize and banana plant again. For the seed germination, all the single as well individual inoculated treatments show improvement, except T5, which confirmed that the AMF proliferation and germination is triggered in the presence of MHB or MHB released molecules. And and they both have a synergistic interaction to improve the seed germination as well as shoot root length and then describe it with phosphate content of the root. While based on the field study on banana plant, again T7, which include T7 showed the marginal improvements by increasing the morphological and biochemical characteristics of the banana plant. But later on T8, T6 and T4 shows the significant improvement, which include the MHB also. Where biochemical parameter also shows 6-fold improvement in T7 followed by T8, T6, T4 compared to the control. The biochemical, these parameters are also improved, while all the biochemical parameters of this like mycorrhizal colonization is also improved, and T7 and T8 shows increased mycorrhizal colonization as well as number of spores and root protein content compared to the control and all the treatments. So here is the chapter conclusion of last chapter, that signaling molecule was extracted and purified from the MHB. The extracted molecule was analyzed and identified through various analytical techniques. The structure of signaling molecule was elucidated after comparing with standard, and results revealed that it is anti-oxohexone C6-homocyryllactane. Among the various treatments, T7, which contains NaHL and EMF, shows better results on morphological and biochemical parameters of the banana. It also promoted spore germination and mycorrhizal colonization as compared to the control. The PLF analysis confirmed that T7 has significantly improved the total microbial biomass compared to the control. These are the key findings and future prospects of the present study, where the present study identifies Staphylococcus aureus as a novel Ph.D. Enterobacter protein, initially identified as MHB, also shows PGPR properties. And among the different combinations of TSD03, MHB and AMF and their different combination, MHB plus AMF shows the best result. However, NHL also shows the good results. The future plan is necessary to standardize the large-scale production of the bioformulation. These are some of the references I have been going during the project. These are the list of posters, papers and oral presentations I have participated and presented. These are the workshops and symposium, and these are the conferences I have been attended. These are the list of publications. Some of them are already accepted and some are under the review. Lastly, I would like to take this opportunity to acknowledge. So I would like to thank Ms. Yushambu for giving me this life and my beloved family for this support. I feel immense pleasure in expressing my reverence and heartfelt gratitude to my guide, Professor R.V. Subramanian sir, for bringing my parents' dream into a reality. And I am incredibly thankful to God for giving him as my PhD guide. Thank you, sir. I am thankful to Professor Anuradha Nerukarman from MS University, Vadodara, for providing culture of CV026 and the GSFC soil testing facility for all the soil testing, soil analysis and their correlation. I am thankful to former and present head Professor K.C. Patel sir and Professor Vibha Trivedi sir for all those regular brainstorming sessions and DRC committee. I extend my sincere gratitude to all the family of biosciences. Many thanks to garden staff, specifically Ajay Bhai Nalkaka, Indravadan Bhai Kisan Bhai, for helping me to carry out the field experiments at Botanical Garden. I am grateful to have Ketan sir and Himanshu for their constant suggestions and troubleshooting during the work. I would also like to thankful to departmental friends and some lifetime friends. Lastly, thank you for the Gujarat government for providing financial support at the show and Sardar Patel University.
It is a wonderful work and comprehensive work which you have been presented, and the nature of the work, the outlook of the work and mass task you have been completed. I have a couple of questions because I have gone through this in nicely and I have given a comment with you. One thing, when you talk about in the beginning about the phosphate utilization thing.
Yes.
So what are the fate after this when you conduct the exercise? So how much it is cover up of phosphate utilization? When we talk
After
about
bacterial inoculation.
Yeah.
So initially I used 20%, and when I added bacterial experiment and finishing all the experiment, then it will convert it to
number one. Number two is that when you talk about only one side, you have selected this particular remaining selection samples.
For
Rhizospheric
sample collection.
Why don't you check other sites where maybe possibility chances that diversity may be more there, you will be able to find. Although the work was excellent, no doubt about it. But maybe you couple side maybe more to chosen. But the reason specifically for that only.
Sir, actually the field which we have selected, that they have experience of 22 years of banana farming. So we are looking for that those we have already from the banana, they have a more potential for this organisms to be survived. Yes.
The molecular work which have been conducted for this microorganism. So how this associated this T2, T1, T8 to this fungi? It's good, or how it's going to be affected? Which is the best options combination you can select?
I can suggest MHB plus mycorrhizal fungus has the best combination, because if we add compound, then we have to add it again.
What is the reason for that? What is the parameter?
They have more compatibility, as MHB release several enzymes, so that can avail of various nutrients which are already presented in the soil, and they have carbohydrate utilization. So if there is a other carbohydrate, they can utilize them and convert it into a form that can be utilized by the fungus also. So the survivability of both the organisms.
Actually, madam का report का काफी support है. Madam has asked several questions.
I have the
You have.
Yeah.
So you have prepared an answer and you have corrected to show it to madam.
Okay.
And if any queries are there, madam will ask.
First, I have why two selected, so I presented that.
Yeah, you did. I noticed.
Yes.
So I don't think you have tried to solve it.
Yeah. We have also corrected several things in the thesis also.
Yes.
As per your suggestions, we have made it.
So how did you prepare the bioformulation?
Ma'am, I selected two materials. One is wheat straw, which is easily available, and the soil, which is artificial.
In what proportion you mixed and what did you do?
Initially, we tried to do it equally, but after certain time, the fungus growth is very much high after some
That
time.
was also going to be
So-
one of my questions.
Yes.
Like how are these two, along with the wheat straw,
No,
going
ma'am.
to be
So
compatible?
later on, what we have did it after bioformulation for there is a 30 days. So after we proportion it like 2:1. So bacterial population is double, the fungus is in the single population. So after growth phase, it will propagate multiple in time.
And they both had similar requirements?
No, ma'am. The fungus require more amount of moisture, but when we add so much amount of moisture in this, so during this study we did it like we grow bacteria first, mix it with the wheat straw. So once they are survive with each other, then we use that particular substrate only for the fungus. So then it will be easy to for both the organism to take up this.
I had also another query that there was a lot of pH fluctuations that we could see in
Yes,
the table. So how so much pH fluctuation is going to be there?
Ma'am, first of all, initially there was a question also.
In one of the tables,
Yes,
was
yes.
a wide range of pH
pH fluctuation. So first, initially the control soil has a pH in alkaline range, and those who we have added the bacteria, they have the pH which is very low, like 4.5, due to the acid production of the growth. But after the bacterial, after the fungus inoculation and after the fungus growth, the pH is again normalized to 6 to 7. So that is the reason.
Organic acid is one of the reasons of phosphate solubilization.
Yes, ma'am.
But still you saw this effect.
Yes, after bacterial inoculation, after fungus inoculation, the pH is again fluctuating from 4 to 6. It shifts from 4 to 6, so it is in normal range after the fungus combination.
Until what step did you do the field study? How many days?
For 90 days.
For 90 days.
90 days.
And
After
fruiting
fruiting stage, we are not there because
in 90 days you get fruiting in banana.
we got flowering till 90 days. Then later on for fruiting we have left it, because after that we used to add potassium for the flowering and all. So,
Okay.
till that we
I'm
have
not aware of. I mean, how, at what stages the banana requires what? So I'm asking this.
Initially actually nitrogen, then for the growth phase and survival it is phosphorus, and once the fruiting is started then they add potassium so that fluorescence and all the bunch will start.
So labs are doing this in their laboratory. This is other labs in this. Like they have tissue.
Just out of curiosity, there are
Yes.
so many tissue culture pamphlets
Yes,
also
yes.
available. Huh?
They are doing tissue culture.
Yeah. So did you ever think of trying it out at that small scale?
Yes ma'am, actually initially we were started with tissue culture plants.
Okay.
Then we left for the plant
Then
field
you
study.
tried plant-
Yes ma'am.
field.
Initially Ph.D and AMF were cultivated in tissue culture only. Then we have shifted to.
So the inoculum and bioformulation which you did was at that step?
Yes ma'am. After MHB addition, we have started with the field study. So that
You got a good nitrogen result also.
result. Yes ma'am.
But you were never aiming for that, right? You never did that. But
In detail,
yeah.
Yes.
It was not your aim.
Yes.
Right.
Phosphorus.
Huh. But still you got a good result. That's nice. Very interesting. It's a good offshoot. Yeah. It was a wonderful presentation. Very good.
So, any questions from audience?
So you have demonstrated quorum sensing
Yes.
for interaction between AMF and MHB.
Yes.
Did you try to validate by using an inhibitor for this?
Sir,
Because that would, I think that would have completed the entire. Because you have shown positively, but
negative.
But it affects really these.
yeah.
Kabhi kabhi.
And in nature there will be so many inhibitor producers,
Inhibitors, yes.
right? Or quenching will be there when you go for actual application.
And the question that Madam asked and that you have observed, obviously when you have phosphate solubilizing organisms and phosphate is to solubilize, pH is to go down. What would be the reason for increase in pH with application of AMF or otherwise?
Sir, in field they are using the nitrogen-based fertilizer. They are alkaline condition actually. So if we all add organisms, they have acid, then it will neutralize automatically once nitrogen. In our study, we didn't use any type of this product
Yeah.
to neutralize, because after addition of AMF, we actually did it in vitro in lab first. So when we add the manually to how this AMF grow in same medium, and then they will start the
Yeah, true. So, nothing to comment on your work, but just as a scientific discussion, when you are getting this type of results, there is one possibility that there must be organic acid utilizing organisms there, and you could have used some of this or the organic acid that is being produced by your phosphate solubilizer as a carbon source to isolate or to show that it is being utilized, and probably that would be the source. That would be the reason for increase in pH subsequently. But if that is so, then how can this pH go so down? And the reason, the possible answer hypothetically can be that the organic acid utilizing organisms initially would not grow at that concentration, but when it comes down to a certain concentration, they start utilizing it as a carbon source. So there is dynamics probably occurring there. It's really interesting to look on those things also. Good work.
I have one or two questions.
Yes.
As far as I have understood, for one of the isolate you have checked pesticide degradation.
Yes, sir, both of the bacterial isolates I have used pesticide degradation.
One was Enterobacter and another was
Staphylococcus.
Staph. So I was curious, why have you checked pesticide degradation?
Sir, actually, all of the 8 pesticides are widely used in banana crop production. So if we are preparing for bioformulation, if this organism also have ability for bioirrigation of pesticide, that could be a good option.
Yeah.
Second thing, if they lower down the concentration for one transition of that compound from higher toxicity to the lower toxicity, then it can be useful by the other organisms. As
Yeah.
nature has
No, I thought it other way. Do paste control on its own? Do they have ability to inhibit pathogens or some?
No, sir.
No. Because generally we check that. No, otherwise if you support the growth of banana and you degrade pesticide, that means you are growing banana for the paste.
No, sir.
And one of the isolate mechanism was organic acid for phosphate solubilizing. For the other one I didn't get. What was
Also,
it?
we have performed the HPLC also, and they are also producing organic acid, but they have more potential to produce acids actually, like acid phosphatase.
Right.
The concentration between organic acid and enzyme production is second is that was confirmed by carbohydrate utilization.
I see.
So if they increasing more amount of enzymes and acids, so they utilize carbohydrates.
Very nice work.
Thank you.
Congratulations for the work. उसके बाद नहीं वो तो after the external we can say.
Any other? Sir, yes.
Yeah, fine. Enough discussion.
PhD में भी बहुत हुआ.
So would you like to share any experience with your colleagues? Like what they must be more careful when they are going about it.
For field study, it is very dependent on the temperature sensitivity and the pest. Specifically for banana, monkeys are the major problem.
Microbe.
Because many times I started with the fruiting stage. Actually, pre-PhD sir told me about the fruiting stage, how much amount of. So whenever we go for counting of that fruiting bananas are first, they come and they collect.
So before you take a
complete.
So your microbes never had any effect on monkeys?
the monkeys flourished even more.
So you should suggest we should have a good greenhouse to facilitate. I was expecting you to say many more things like you had so much of like such a nice model to work on, and if you had access to confocal or such type of technologies, this would have been still enriched,
Yes.
right? Because the type of interaction studies that you showed, if this could have been shown with confocal, it would have been more, right? Yeah, we want you to continue this in PDF or for your future work.
But is PDF is on this? No.
No, sir.
Cancer
Nematology.
Mm-hmm.
Nematology.
But related to agriculture.
Yes, sir. Potato cyst tomato.
Good, that's a good achievement.
Yeah.
And before doing your
Continuing,
getting your degree.
So it's up to you. Both are examiners.
I will congratulate you for successful defending thesis. So all the best.
Congratulations. Congratulations and all the best for your future.
Thank you.
2 students more to go.
Yeah,
Congratulations to Subramanian sir also.